Hi, I am Achinto Kundu, worked in Center for Molecular Spectroscopy and Dynamics, Institute for Basic Science and Korea University. On behalf of all my co-authors, Dr. Pramod Kumar Verma, Professor Minhang Cho, I am pleased to present our recent work entitled How Molecular Crowding Differs from Macromolecular Crowding, a femtosecond mid-air pump-up study. Crowding is an inherent property of living system in which biochemical processes occur in highly concentrated solutions of various finite-sized species of both lower molecular weight molecular crowding and higher molecular weight macromolecular crowding. The effect of crowding on biomolecules such as protein, enzyme and nucleic acid have been studied in great details, such as in this reference in this slide it was observed that the structure of valine proteins become increasingly destabilized upon increase the Crowder concentrations via protein-protein interaction. However, it should be noted that these changes in protein structure and stability induced by crowding agent are likely to be correlated with water structure, but unfortunately there do not exist many reports on time-result vibrational spectroscopy investigations of water dynamics in crowding environment. So far, researcher has been using different type of polymers such as dextran polymer, glycol and polyethylene glycol as a crowder and mimic the cytoplasmic environment. These molecules are used as a crowder as these are inert and highly soluble in water. We are interested in studying water hydrogen bonding dynamics at the crowded environment. Last year, we used mid-air pump-up spectroscopy to study the water dynamics in macromolecular crowding environment. The model crowder that we choose to investigations is PEG-DME. Figure A shows the representative isotopic IR pump-up spectra of OD stretch mode of HOD in 88% of PEG-DME solutions. Yellow color is due to bleaching and stimulated emissions and green color is due to excited state absorptions. We got the vibrational lifetime of OD stretch mode of HOD in different weight percentage of Crowder as shown in figure B and C. These two vibrational lifetime is due to two different environment of HOD molecule. One is water interacting with other water, that is OD water component, and other is water interacting with oxygen atom of PEG-DME, so that is OD ether. One of the most important observation is that the decay time of the first component, that is OD water component, is negligibly depend on Crowder concentration. Cytoplasmic environment contain both the molecular and macromolecular crowder. A question that remain unanswered, however, is how molecular crowding affects structural and dynamical property of water and how these differ from macromolecular crowding. To answer this, we used the molecular crowder DEG-DME, which is smallest unit of PEG-DME. To gain a local and microscopic picture of crowded system, we used three different IR probe shows here and employed femtosecond mid-air pump-up spectroscopy. One of the three probe is water itself, that is OD stretch vibration of HOD that provide information on the local structure of the water from the perspective of water. The other probe are azido stretch mode of both hydroxylic acid and TEG-MEN3. The former provide information from the solids perspective, while the latter is used to determine how Crowder itself respond to the crowding stress. Here shows the FTIR spectra of these three IR probe in different weight percentage of molecular Crowder. Figure D, E and G shows the peak positions and compare with macromolecular Crowder. F and H shows the full width hub maxima and compare with macromolecular Crowder. Molecular crowding causes a large blue shift of OD stretch IR spectrum. The large blue shift indicate not only a redistribution of equilibrium hydrogen bond number and hydrogen bond strength, but also the HOD molecule tightly associated with ether oxygen of DEG-DME. Unlike the OD band, the azido stretch mode of both solute and Crowder undergo substantial redshift with increasing Crowder. As crowding increases, the water-ether populations build up at the expense of water-water populations and HN3 encounter fewer water-hydrogen bond donor. What is more interesting is the increase in full width hub maxima of azido band of HN3 up to 58% DEG-DME, followed by a decrease further increasing crowding. This increase in full width hub maxima until 58% indicate the presence of heterogeneous hydrogen bond environments around the azide group. However, when the crowding become extreme, that is more than 58%, the solid HN3 experience mostly water poor environments. This disruptions of water hydrogen bond network is further confirmed by observations that the peak positions of azido stretch band of TEG-MEN3 in highly crowded medium reach a value that is close to the TEG-M3 in dichloromethane. The full width hum maxima of TEG-M3 gradually increase with crowding but does not show any sharp transitions like PEG-DME. These sharp transitions of full width hum maxima for PEG-DME exhibit strong crowding dependent conformational transitions at macromolecular crowding of around 68%. The vibrational lifetime of the IR probes depend on the surrounding environments. We got the vibrational lifetime by doing the mid-IR pump-up measurement. Here we show one of the representative IR spectra of OD stretch mode of HOD in 9 to 8% DEG-DME. The yellow color is the bleaching and stimulated emissions, and green color is due to excited state absorptions. We use the kinetic model, one decay component below 38% DEG-DME and two decay component above the 38% of DEG-DME. These decay components are shown in B and C, and the corresponding vibrational lifetime are shown in D and E. In dilute solutions, populations decay is mono-exponential with time constant similar to that HOD in net water. When crowding is increased to 38% and above, the populations decay no longer fit to a mono-exponential function but instead fit a bi-exponential function. The faster component corresponding to OD water and slower component corresponding to OD ether. The vibrational lifetime of the OD water component gradually increases with crowding, and the increase in lifetime is significant after crowding of 58%. Similar, OD ether populations exhibit a marginal increase in lifetime value up to crowding of 58%, after which the lifetime becomes much longer. Thus, crowding greater than 58% strongly affects both the OD water and OD ether populations and significantly slow down their vibrational relaxations. Carefully observations of the peak positions of OD water component also reveal that the peak position is marginally blue shift until 58% crowding, and crowding more than 58% dramatically shift the peak positions to a higher wave number. Compared with PEG-DME in less crowded media, molecular and macromolecular crowder behave similarly. In highly crowded media, molecular crowder significantly slow down the dynamics of both OD water and OD ether component. In contrast, macromolecular crowding does not affect OD water but moderately slow down OD ether component. The vibrational probe covalently attached to the molecular crowder provide a direct assessment of crowder's behavior. Figure A, B, C shows the IR pump-up spectra of azido stretch in dichloromethane, 10-8% crowder and 98% crowder. The blue color is due to bleach and stimulated emissions, and yellow-red color is due to excited state absorptions. In dichloromethane, the excited azido stretch vibrations in TEG-MEN3 decays with two time component, 1.3 picosecond and 6.5 picosecond. In dilute solutions and less crowded media, both fast and slow time constant accelerate, reflecting the sensitivity of the azido stretch mode of local water's density. At increased crowding, both lifetime component become progressively slower and reach the value that close to that TEG-MEN3 in dichloromethane. This increase provides important evidence about the local water density surrounding the TEG-MEN3. To determine how crowding affects a water-sensitive solute, the azido stretch mode of HN3 was used. Figure A and B shows the IR pump-up spectra of azido stretch mode of HN3 in water and 98% DEG-MEN3 solution. The vibrational lifetime of HN3 is plotted in figure C and D as a function of molecular crowder concentrations. As the crowding increased from 0 to 58%, the lifetime of azido stretch vibration gradually increased from 2.5 picosecond to 3.9 picosecond. The first vibrational relaxation remain mono-exponential until 50 8% crowding indicates that DEG-DME first and second hydration cells are not very different from bulk water in less crowded medium. The increase in lifetime until 58% is due to disruptions of water extended hydrogen bond network and heterogeneity in hydrogen bonding. DEG-DME is the smallest of PEG-DME based polymer, thus it cannot form highly extended structure hydration cell like PEG-DME. Consequently, at crowder of 7 to 8% and above, intramolecular effect dominate and molecular crowder are forced to share their hydration cell to cope with increased crowding stress. HN3 lifetime is no longer exhibit a single time constant, rather has two different time constant. In summary, the use of three different vibrational probe and femtoseconds mid-air polarization sensitive pump of measurement offer detailed insight into three important constituents of intercellular environment: water, crowder, and water-sensitive solid molecules. The most important finding is that molecular crowder affect both the OD water and OD ether in highly crowded media, but macromolecular crowder does not alter OD water and has only modest crowding effect on OD ether component, even in highly crowded media. PEG-DME preserve water hydrogen bond network by adopting a compact helical structure. For DEG-DME, due to its low molecular weight, DEG-DME facilitated water-mediated cross-linking and subsequent water aggregations in highly crowded media. Our experimental data support that water in highly crowded solutions like in cytoplasm will be significantly affected by high molecular crowder but exhibit bulk-like behavior in presence of high macromolecular crowder. Finally, this work was supported by IBS, project code IBS R023D1. All IR pump of measurement were performed by using multidimensional spectroscopy facility in Seoul Center of Korea Basic Science Institute. I would like to thank all of my lab members. This paper is dedicated to the memory of our wonderful colleague Dr. Pramod Kumar Verma, who passed away recently. Finally, I would like to thank everyone for your attention.